Hi, everyone. Welcome to the Johns Hopkins Cerebellum Seminar Series. Today, we have David Schoppik. David studied smooth pursuit eye movements for his PhD and was mentored by Steve Lisberger at the University of California, San Francisco. There, he discovered that in the frontal eye field, populations of neurons together have responses that predict eye velocity fluctuations. Then, as a postdoctoral fellow in Alex Shear's lab at Harvard, he developed a zebrafish as a model to understand the vestibulocular system. Working with Bob Baker, he discovered that even through larval zebrafish, the fact that they lack semicircular canals, they have a reasonable VOR, and he figured out how the vestibular neurons make this possible. He's now an associate professor in the Department of Neuroscience and Otolaryngology at New York University School of Medicine, where he's built a world-class zebrafish lab that studies control of gaze and posture and the role of the cerebellum in those functions. Good to have you, David.
Thank you so much, Reza. It's a pleasure to be here. I can see a couple friends in the Zoom participants, and it's a treat. Thanks so much for coming. So I feel this is a special audience because of the cerebellar focus, and I want to just share a little bit about where I'm coming from and what the cerebellum means to me, right? Reza and I went back and forth about this, but the truth is that the smartest trainees in Steve's lab studied the cerebellum, and they studied its roles in motor learning and made all kinds of beautiful progress and discovery. And so those were the people that I looked up to. And indeed, the cerebellum as a structure and the rich history of trying to understand contributions to learning have motivated me throughout my career. So I have always had it in, no joke, the back of my mind. And I've thought often about what we could use our system for and how we could let the things that we're interested in intersect with what we think the cerebellum is doing. So in some sense that it's been a little bit disappointing because over the past 10 years, we don't have a beautiful, this is what the cerebellum does. And I think it's important as we go through this talk to understand why. And mostly the why comes from my initial thoughts about circuits. So as a student, as a PhD student, neural circuit was something that you could get delivered. FedEx would drop it off at your door, and if you just poked it and prodded it and eavesdropped on the right parts, you would be able to infer what every part did. And then as a postdoc in a developmental biology lab, I came to appreciate that it wasn't that way at all, that this was something that had to emerge, that had to be built. And bringing those two ideas into line to understand how development constrains what circuits can and can't do has been the conceit of the lab, and that's led us near to the cerebellum. And I'll share a work in that space here today. So the title of the talk is What the Cerebellum Does and Doesn't Do. We'll do that in reverse order and start with doesn't and then move on to does. The lab studies balance, how it develops, functions, and fails. And today we'll talk about develops and functions. Fails is for a longer audience. So let's start with how it develops. We use the larval zebrafish, as Reza said, as a model. The things you need to know about the fish for this talk are that they're small vertebrates. The balance and gaze stabilizing brainstem circuits are evolutionarily ancient. So before there are trees on land or bugs flying in the air, or even before Saturn has rings, there are sharks swimming around using the same brainstem structures to process gravity and guide posture and gaze control behaviors. The zebrafish, like other vertebrates, has 6 eye muscles that control each eye. There are 3 cranial nuclei. There are, and this is the fun part, only 175,000, 200,000 neurons total that we have to worry about. So it puts us sort of on the scale of a Drosophila. I like that. I like countable numbers of things. The fish are transparent, so they're very amenable to all of the advances in optical microscopy. They're also genetically accessible. So a lot of the things that we can do rely on labeling particular populations of neurons. For the impatient among us, which is certainly me, and I suspect most of you, it's really neat that they develop very quickly. So from the time sperm meets egg, it takes them 4 days till they're swimming freely. And then lastly, for the first part of the talk, it's important to know that their photoreceptor mosaic is topographically organized. So that is a fancy way of saying it really ought to stabilize their gaze as they climb or dive in the water to take advantage of all of that retinal specialization. This is Paige's, this is 1/3 of her PhD thesis. Paige is now postdoc in Felice Dunn's lab in San Francisco. So Paige studied the conserved circuit for gaze stabilization, which we'll quickly review, but should be familiar to most of you. There are gravity sensors in the inner ear that relay information about head or body tilts via peripheral afferents of the 8th nerve to central brainstem interneurons that transform those signals into commands for the motor neurons to move the eyes. Okay, here's a fish doing it. I'll play the video. Don't worry if it's choppy. The main thing is in the upper right, you have an image that's zoomed in of the eye, and you can watch the iridophores, those shiny white lights, reflect white dots reflecting the infrared illumination as the fish swims in the dark. And of course, you can see the gaze stabilization. No, of course you can't because it's much too fast. So I'm just going to superimpose 4 frames. You can see the body of the fish changing from nearly horizontal to considerably nose up. And in the bottom, there are 4 frames where you can see the eye, though that corresponds to each of the body positions. Note that the little white dot that's at about 2 o'clock is the same IR reflecting cell in the same orientation relative to the horizontal, which says that although the fish's body has changed orientation, the eye relative to the horizon has remained at the same orientation. And that is how the fish stabilizes gaze, and indeed how we do. So muscle names don't really matter, but there's a pair of muscles, superior oblique and inferior rectus, that rotate the eyes in the torsional plane when those are the pulling directions, the arrows. There's an opposite pair that pulls the eye the other way when the fish dives down and rotates the eyes up. Okay, the main finding of the first half of the talk is that the gaze stabilizing reflex can mature without visual or vestibular feedback. So how do we evoke the vestibulo-ocular reflex in the dark as experimentalists? Well, we embed a fish in essentially a jello shot, and we put that jello shot on a table, and we can rotate the table. The fish can't swim away, but we free the eye so we can measure what the eye is doing. The velocity profile of the table and position of the table is matched to steps that the fish perform normally as they're swimming. So this would be a 15-degree nose up step with a trapezoidal velocity profile that peaks at 35 degrees per second. And in this case, the right eye of the fish should rotate counterclockwise. So what Paige found, confirming what we'd seen earlier, is that gaze stabilization improves across early development. So here's the responses of the fish as a function of time. You're going to see velocity on the left graph. The dotted line is at 35 degrees per second. So the fish is trying to match the velocity of its eyes to the velocity of the table. And then by convention, the ratio eye to table is defined as gain. So gain of 1 on the right side would be perfect cancellation, perfect performance, and that's the dotted gray line. And what you can see at 3 days post-fertilization, either as an average trace on the left or as individual circles for individual fish performance, there's not much of an eye movement at all in response to that table step. But progressively, as the fish gets older, 5, 7, 9, and 15, the fish is able to match its eye velocity to the table velocity in this paradigm. And so we say that behavioral performance improves over the first week or so of life. You can see they're real good at 9, almost asymptoting, not getting much better after 10, 11 days. Okay. So is that maturation that you're seeing, that improvement that Paige recorded of the vestibulo-ocular reflex, limited by sensory feedback? And of course, the reason that we thought that this might be so is because of the canonical story involving the cerebellum, where feedback shapes the mature vestibulo-ocular reflex. So the challenge for all of us as we get older, our muscles get weaker, our vestibular system, the hair cells die and performance decreases. How is it that we can maintain stable gaze at 40 or 50 in the same way that we did at 20? And the answer is, of course, that the cerebellum compares the tilt sensation from the vestibular system to the visual feedback that it observes and uses the output of that comparison to adjust the gain of the synapse between the peripheral afferents and the central interneurons. And that is this beautiful motor learning story. And so that's where I was all set. We were going to find that the cerebellum during development, development was just a privileged form of motor learning because you have to learn. So surely the cerebellum will do the same thing. And Paige did an experiment to test that, which is to ask about how the vestibulo-ocular reflex develops in congenitally blind fish. Same traces that you've seen before, except that green is blind and black is sighted, siblings. And you can see that the blind fish at the same age have the same performance. And so that was kind of a head scratcher because they don't need the visual feedback to get better at the vestibulo-ocular reflex. So it's not the classic story involving the cerebellum, because indeed it's not visual feedback that limits the rate of the development of the vestibulo-ocular reflex. Okay, what is the rate limiting process in the development of this circuit? So we borrowed a term from chemistry, where in a multi-step chemical reaction, one of the steps is the slowest, and so determines the rate of the overall reaction. That's why we call it rate limiting. I thought for sure that the rate limiting step was going to be in the sensory periphery because there are so many stories and Nobel Prize given out for the observation that perturbing sensory feedback can perturb adult performance. So I thought, well, that'll be the slowest part. That's where it's going to be. Paige, bless her heart, argued that it would be the motor periphery because why not? And so what we decided was to start by examining the convergence point. So every single thing that happens in the zebrafish brain has to go through the motor neurons in order to produce behavior. So whatever is happening upstream ultimately funnels through this bottleneck. So if we look at progress in development there, we're going to be able to infer something about the nuclei and nodes upstream. So let me just give you a conceptual timeline of how this rate limiting experiment might work. Let me start by saying that the lab knows a lot about the anatomy and development of the circuit. So all the neurons are there, all the motor neurons are there, everything's wired up appropriately before the behavior starts. So that's the papers in the lower left corner. But that gray triangle is meant to schematize the maturation of behavior. It happens after the motor neurons finish targeting muscles. So that's behavioral improvement. So the idea is if we see some part of this circuit mature early, then it can't be rate limiting. The hallmark of a rate limiting step is something whose time course matches the time course of behavioral improvement. And that's what Paige went looking for. And she used a technique called tilt-in-place microscopy that we developed to be able to measure the responses to body tilts without having to tilt a 2-ton microscope. Grant me, in the interest of time, that we can discern down and up responses, and we can geek out about this later. The other thing that Paige had to develop was the ability to track the same exact neurons across time, which she did. You can see an example set of images here for the same 3 motor neurons at 3, 5, 7, and 9 days post-fertilization. So we can also geek out about how she did it later, but what I'm going to plot for you is the motor neuron response strength in the same neurons measured when the fish was 3 days post-fertilization against 5 days post-fertilization. And I'll plot the unity line. So cells that respond more strongly at 5 than they do at 3 to the same tilt will be in the upper left. And indeed, that's what you see. The different markers reflect cells that came from different fish. So we can turn that into a distribution by subtracting 3 from 5, and you can see it's off 0. And to our surprise, between 7 and 5 and 9 and 7, the average change isn't high, it's 0. And so the changes are sort of stopping between 5 and 7 before maturation. And that implies that the rate limiting step is likely downstream of motor neurons. So certainly pretty far from the sensory periphery, as I'd thought. Now, Paige is an excellent scientist, did a bunch of controls. It's true for the central vestibular neurons. It's true for other measures of functional development, like directional selectivity. It's true for other pools of motor neurons than the one I just showed you. And it's even true in fish that don't develop ocular motor neurons. Everything that's going on in the brain seems to mature before the behavior does. So what's downstream of motor neurons? Well, Paige went and looked at the muscle. So you can see the striations on the left at 3 days post-fertilization. That's the superior oblique muscle. And what you can't see, because there aren't any there, are the black alpha-bungara toxin stains. But you can see them emerge between 5 and quite strongly at 9. So this is the aggregate of acetylcholine receptors in the muscle that define the neuromuscular junction. And that sure as heck seems to be developing nicely. Paige then checked, are the alpha-bungara toxin, are they actually opposed to the vesicles in the motor neurons? That's the SV2A staining in the middle in F. You can see them superimposed. These are indeed the neuromuscular junctions. And so Paige could quantify the development of the neuromuscular junction. And that's what she did for both the post, using the alpha-bungara toxin that stains acetylcholine receptors, and also the presynaptic vesicles. And so what you can see is that the postsynaptic acetylcholine receptors seem to aggregate with a time course that's very, very similar to the development of behavior, where there's not a whole lot at 3. You can see slow progressive increases 5, 7, and 9, and then it asymptotes between 9 and 14. Contrast that with the presynaptic vesicles in the motor neurons that are some a little bit there at 3, and then indistinguishable 5, 7, 9, and 14. So to put us back in that framework, gaze stabilization behavior improves. I've shown you that the motor neuron response matures earlier. I've asserted that the central vestibular neuron responses also mature, and the neuromuscular junction seems to develop with a time course that matches that of behavior. So we can test if vestibular sensation is rate limiting now by using fish that are transiently gravity blind. So this is the idea. First, I've got to tell you how we make gravity blind fish. They're a mutant that doesn't have the otolith stone on the utricle, which is the primary gravity sensing organ in the zebrafish. You can see that by comparing what's in that oval on the left and the right. So some of those fish, a small number, will actually redevelop that utricular organ when they're 14 days, 13, 14 days old. And so that means that the entire system develops absent any kind of gravity sensation. So there are two possible outcomes for those fish. Paige reasoned by asking what happens when the fish is 14 days old and gravity sensation begins, she can tell if gravity sensation is dispensable, because if you didn't need gravity sensation, then the vestibulocular reflex performance at 15 days, a day after gravity sensation started, would be ready to roll. Contrast that if you needed gravity sensation to tune the reflex in some way, then we would need that same 4 or 5 days of behavioral improvement to happen. And so what Paige found was that immediately after restoration of gravity sensation, the gaze stabilization behavior was mature. So you're comparing red and black now, the siblings, the autogellin mutants that were delayed, so their gravity sensation came on at about 14 days, and then their siblings that never developed the utricular otolith that have almost no measurable response to body tilts at all. So the main finding is that the gaze stabilizing reflex can mature without visual or vestibular feedback. The inference, of course, is that the cerebellum would be dispensable then, even if that's this lovely comparator organ for visual and vestibular feedback. So we propose that the development of the neuromuscular junction is actually the rate limiting process in the maturation of gaze stabilization behavior. So let's zoom way out to end this part and just ask, what can we infer by considering the rate limiting process? Well, the basic biologist answer is that evolution ultimately has to regulate the rate of circuit development. So even within the zebrafish, spontaneous coiling, motor behavior happens just 1 day after sperm meets egg. So well before the vestibulocular reflex, spinal acetylcholine receptors are in place because the tail moves. And the very first sensory motor behavior that zebrafish show happens well after that. You can see a touch-evoked escape response at 48 hours. So there, the neuromuscular junction, the entire development of the motor system can't be rate limiting because they're moving before they can do evoked movements. So the rate limiting process for another sensory motor behavior, this touch evoked escape, has to be upstream of the motor periphery. There's a natural experiment that's been going on for many millions of years where altricial animals, think, rodent pups, think dogs, or our kids, Ruby and Joey, they can't locomote at birth. They're sort of luggage. They stay where you put them. You can contrast that to precocial animals, which tend to be delicious, like lamb and deer and chickens. Moments after birth, they're ready to run. And they have to be, because otherwise they're going to be eaten. So empirically, when the pressure is on to swim or run or stand or be eaten, the motor periphery seems to develop much more rapidly. Examining the rate limiting process, right, as an experimental approach can give insight into past pressures on maturing neural circuits. And that's the end of Paige's story and the end of the how it develops part. And you can see the cerebellum, that's the doesn't. All right, let's switch gears. Again, in the interest of leaving time at the end for questions, I'll just plow through and talk about how balance functions. So this is Franziska Auer's story that was recently published. It builds on earlier work from David Ehrlich, who's running his own lab at Madison. I believe that you heard from him earlier this year. So Yay. He's doing great. Okay. So again, driven to understand what the cerebellum might contribute to posture, Franzi started by developing a new tool that would allow us to do chemogenetic activation and ablation of cerebellar Purkinje cells. So on the left is a schematic of the zebrafish. In the middle is a lovely microscopy image of the Purkinje cells. And you can see their axons as well, descending from about the middle of the image down. Those are the vestibulocerebellum projections. The little black dots are expressing a fluorophore, or a red fluorescent protein, and also a channel, the rat TRPV1 channel. So it's a cation channel, and it's not found in zebrafish. There's a TRP channel in zebrafish, not this one. And the important thing is that this rat TRP channel is sensitive to capsaicin, the ingredient in hot peppers that's spicy. And zebrafish endogenously are insensitive to capsaicin. So they could swim in the hottest of hot sauces and not be bothered at all. But once we put this TRPV1 channel into select neurons, in this case, the Purkinje cells of the cerebellum, using the ALDO-CA promoter, a low dose of capsaicin should increase their activity, and a high dose should be excitotoxic, in this case, 1 and 10 micromolar, which will denote with yellow and red colors. So let me show you what Franzi did to check if indeed we could see activation and death. So this is the same transgenic zebrafish. It's one image from half of the cerebellum. Each of those circles are the Purkinje cells. In the dotted square are 3 of them, and they're also expressing a nuclear localized calcium indicator. So the bluer that the image is, the stronger the fluorescence from that nuclear localized calcium indicator. So Franzi's going to do a long-term imaging experiment where she's going to put a low dose of capsaicin on and see if there's persistent elevation of the calcium signal. And that's what she could see in the stack of images that have just appeared on the right of your screen. You can see that one of those cells, indicated by the little heart, gets bluer and stays bluer for hours after a low dose of capsaicin. This is stochastic. Not all cells respond exactly this way, and not all cells respond for this long, but a small dose is not excitotoxic because the cells, you can see, are healthy. Well, the zebrafish folk in the audience can see that that's true. But let me show you what it looks like when they're not. And Franzi adds a much stronger dose of calcium, capsaicin. So you've got a little matrix. On the top are 2 images of that are the control. You can see the 7-day cerebellum and the 9-day cerebellum in the same fish. There are more Purkinje cells between 7 and 9. On the bottom, you can see what happens when you leave the fish, starting just after the image was taken at 7. You could see that the Purkinje cells look awful in the lower right. And even the ones that remain, you can see the neuropil is really severely degraded. So if we compare the Purkinje cell numbers, because Franzi went in and painstakingly did that quantification, you can see a plunge if you give 10 micromolar of capsaicin compared to just leaving the fish in the DMSO, the control solution. Okay, so I hope I've convinced you that we can activate and ablate the cerebellar Purkinje neurons with a stimulus that doesn't perturb other fish. Okay, Franzi's going to look at behavior now using a scalable apparatus to measure posture and locomotion, or sample. Sample is pretty simple. It's just a cuvette that's illuminated with infrared light and a machine vision camera that analyzes and extracts kinematic variables as a function of time. So the raw data looks something like this. You can see the schematic of the fish, just to give you a sense of size. We have a bunch of fish swimming around in a cuvette, and the apparatus records video, in this case of the fish swimming from the left to the right. You can see it orients its nose up, thrusts with its tail, and it actually climbs here. We'll do this sort of as a follow the bouncing ball video. We define pitch as the angle between the long axis of the fish and the horizon. So on the top, you can see swim speed, and on the bottom, you can see the change in pitch as the fish climbs. The fish swim in those discrete saltatory bouts that produce translation, and it's often accompanied by changes in pitch. So this is the sort of raw data that Franzi's got to work with, and she can extract all kinds of kinematic variables, speed and angle related. Okay, so her protocol for activating and ablating the Purkinje neurons and doing behavior activation was low dose of capsaicin for 6 hours given twice. And ablation was just an hour of hitting those Purkinje cells with 10 micromolar capsaicin. I illustrate this for you not because the details are particularly important, but only to say that it took a little while to work out exactly how to do this. But we did. And so now let's see what the consequences are for fish posture. I'm going to, again, in the interest of time, gloss over a whole bunch of things and just highlight that we can see similar phenomena when we perturb the cerebellum and we look at the posture that the fish maintain when they're climbing. So remember that we're doing yellow activation, red lesion. When we activate, we can see that fish climb with a more nose-up posture. When we ablate, you can see that fish continue to climb with a more nose-up posture. We're disrupting the sensation of body relative to the horizon when we perturb, either by activating or ablating Purkinje cells in the cerebellum. We talked a little bit about development. Here's the striking example set of images that Franzi had from 7 days and a week later at 14 days of the same fish. They give you a sense of just how much development of the cerebellum there was over this period. So are the consequences comparable? And for climb bouts, the contribution of Purkinje cells to those climb postures persists across early development. You can see 7 days, there's a increase in posture, a comparable increase in posture at 14 days. So it's not like you get a bigger cerebellum and then the Purkinje cells seem to matter less or they seem to matter more. They're still playing a comparable role in maintaining the posture of swimming fish. All right, we focused on climb bouts. Why are we focused on climb bouts? I'm going to tell you a little bit about what fish use those climb bouts for. They navigate up and down in the water column. Here's a video that shows you an example of how they do it. So they'll rotate. They'll have these sequences of bouts with a matched trajectory, and then they'll reorient towards the horizontal at a different elevation. They can't, with what thrust they can produce, go all the way up as high as they'd like. So they need to sequence these bouts, and they need to do certain things during those bouts to actually get the elevation that they want. Let's talk about that for a second. So they have 2 ways of gaining elevation that have to do with the effectors. The first, of course, is the trunk. That's the primary means of thrust. They undulate their tail back and forth and propel themselves through the water. They've also got paired appendages, just like you and I have arms. They've got pectoral fins, and those fins might be able to produce lift. And that's what David Ehrlich found in his 2019 paper in my lab. Let's talk about the modes of locomotion. If you consider a rocket, the rocket goes where its nose is pointed. And so too would a fish whose climb is completely dominated by its thrust. The nose points where the fish goes. So if you plot the posture of the fish against its trajectory, you would expect them to sit along a line. And at younger ages, at 1 week old, that's exactly what you see. But for a small number of points that lie off the line for any given bout, for many, many fish, the fish kind of swim like rockets, especially when they're young. Contrast that with what a helicopter can do. A helicopter can achieve lift and go up in the air without reorienting. So it doesn't have to tip its nose up to go up. Maybe fish can do that. Maybe there would be a difference between the trajectory and the lift produced that would imply that something was producing a change in elevation. You can see in these close-up videos, the fish can indeed move like a helicopter. If you watch the fins in this one, they're time-locked to that increase in elevation. It's real tough to get videos like this, but this is how we know what's going on. And also from an experiment like this, if we look at the older fish, same graph that you saw, posture versus trajectory. Here, the points are off a line. But if we simply pull the fins off, we're going to lose the dots that I point to with the arrow. This is the control data, and this is a finless fish. So you can appreciate that the difference between trajectory and posture, that additional lift, is due to the fins. And David, in his paper, looked at how larval zebrafish climb. The two things that they do is they rotate their trunks and generate thrust, and they also, he found, use their pectoral fins to generate lift. He did two more experiments in this paper that set the stage for Franzi's work. He found that gravity-blind fish can't, even though they can move their fins and they can move their trunk, they don't coordinate those two, implicating the vestibular system in that coordination, and had a much cruder assay of Purkinje cell function, but showed that this loss might disrupt coordination similarly. So that was cool. And that led Franzi to ask, what about the cerebellar contribution to this coordinated behavior? So now she's looking at the lift produced and asking what happens when you lose the cerebellum. Franzi had one really interesting observation that was very powerful for interpreting this data, which was that the amount of lift, if we're measuring that as distance, is going to be speed dependent. And so in order to really understand this, she had to break the data out by speed. You can compare, now we're plotting the upward rotation against the lift. It's similar to the graphs that you saw before of posture and trajectory, and breaking this data out into the fastest movements, the slowest and medium speed movements for both control and Purkinje cell lesioned fish. And what you can see is that the slowest swims aren't affected, but when you take speed into account, the faster climbs, where you would see measurable changes in lift, are impacted by loss of the cerebellum. So the cerebellum seems indeed to contribute to this coordinated fin and trunk movement, and it's really apparent when you break the data out by the speed of the bout. Cool. Does the cerebellum encode posture? This is where I hope most of you are rolling your eyes, because duh, of course it does. There's so much data on this, but it's important that we ask not just does it, but what kind of signals do we see when we destabilize the fish that might give us really fodder in the future for the sort of models of cerebellar development and postural encoding that we want. We're building here on lots of work by other labs, Indira's lab, even Aaron Schumann's lab, and recently beautiful work by Laura Nogler in Ruben Portugues's lab, did really a survey of the responses of cells in the larval zebrafish cerebellum to all kinds of motion stimuli, nicely broke it out and showed that the vestibulocerebellum was likely to be the lateral Purkinje cells that were also involved in eye and body coordination. I recommend this paper highly because it's really, really nice work. And we built right on it, looking at Purkinje cells out on the lateral parts of the developing cerebellum. So Franzi's going to use that same TIPM microscopy to ask if the cerebellar Purkinje cells were tilt sensitive. So if we tilt the fish and look at the responses of Purkinje cells, what do we see? She gave the same cells nose up and nose down tilts, in this case, that were 30 degrees, so quite large. The line that we had to measure Purkinje cell activity was quite sparse, which was good. It was easy to identify individual cells, but I don't want to give you the sense that this was in any way a complete survey of what we saw, nor were we measuring dozens and dozens of cells simultaneously from the same fish. So we're limited. This is our initial foray here, but it was interesting. I'll share it with you. Some Purkinje cells, we were happy to report, are indeed tilt sensitive. So the cell on the left, in response to 20 or so presentations of a nose down stimulus, always responds very strongly and doesn't respond strongly at all to a nose up stimulus. Contrast that with the cell on the right, which responded in that there were measurable changes in fluorescence above baseline, undoubtedly, but didn't seem to be directionally selective. It was not tuned to nose up or nose down. It was just responsive generically. We split the data into tuned cells and not tuned cells to look and see if there was any kind of topography. There didn't seem to be any obvious topography to where those cells were in the either 7 or 14-day cerebellum. So we didn't have new and old, or we didn't have an easy way to map those out. And then we checked untuned cells were just as active in terms of their fluorescence. So the calcium flux was just as great as tuned cells. You're plotting the average up response against the average down response. Tuned cells are gray and they sit away from the unity line, as you would expect. That's why they're tuned. And not tuned cells sit along the unity line, but generally you see just as strong responses. So we're not looking at newborn untuned cells that can't flux calcium for some reason. So Franzi looked at the untuned cells and noticed something interesting, which was that the untuned cells had quite variable responses to the identical body tilts. So you're going to look at a heat map now. You're going to see trials on the horizontal and cells as columns. I'll show you the up trials. Sorry, I said that wrong. Rows are cells, columns are trials. The same cell can respond mostly the same or a little bit more, maybe a little bit less on a given trial. It's true for both up and down. And so we looked at that and said, I wonder if there's something in the not tuned cells. So trivially, if we looked at the tuned cells, we could tell which direction the fish was tipping. We didn't need to really analyze that, but it was interesting to see this variability, to see, could we extract anything from the activity patterns? Okay, so Franzi did first a little dimensionality reduction. So this is principal component space. We're just looking at the responses of the untuned cells, and we've just asked how they respond on nose down trials in that space and nose up trials, because we don't know what features of the time course of activity are important, but maybe they're different for nose up and nose down. So that was pretty promising. It did look like there was information that could be extracted from nose up trials and nose down trials in this low dimension space. And so Franzi did a little bit of modeling. She just built a little support vector machine that asked about decoding accuracy as a function of how many of the cells. Again, they're not recorded at the same time. They're just trying to ask about something generic in their activity that would tell you down from up. So it's a really easy decoding task, but we're looking at what we would call untuned cells. And what you can see is that as the population size increases, each dot is a different draw from the 30, 40 or so neurons that she had recorded from. You can see that our decoding accuracy gets better until it's perfect for a population size, again, interestingly, of untuned cells. Talking to an audience of aficionados, I hope that you don't find this surprising or shocking, that in the collective activity of Purkinje cells, information seems to be carried about body posture. It's just neat that we can measure these sorts of things in the larval zebrafish as a precursor to doing more sophisticated analysis with better reagents. And I hope that's the message that you take away from the imaging. The behavior is nice because it tells us that, yes, indeed, the cerebellum is probably doing something. And it's worth looking at the activity patterns of the cerebellum to see how those might explain the behavioral changes after loss that we see. But it's really first steps in a larger and longer program. Zooming way back, I've told you a little bit about our studies into balance. I've told you how it develops and what we can learn from studying a developing organism about constraints and evolution. And I've told you an initial foray into looking at the cerebellum, how it contributes to posture and how it contributes to coordinated locomotion. So with that, thank the other members of the lab. And yeah, there's a cute picture of a dog. Get you all on my side before the questions. Thanks to my support, and thank you all so much for listening.
Yes.
I'm going to stop sharing.
That was wonderful,
Any
David.
questions?
That's wonderful. Thank you. Thank you so much. It was didactic and so clear. So please, if you have questions, just raise your hand and I'll be happy to call on you. I was thinking about development in humans and how it takes years for children to gain dexterity, and that's thought to have something to do with myelination of the axons from the cortical spinal tract. In your zebrafish, you talked about the neuromuscular junction. Is myelination a part of this as well?
I need the audience to know now that I did not set that question up. That is not a plant. But Reza, you could have no way of knowing that Franzi, who did the second half of the talk, came to my lab after having done a PhD looking at myelin and said, the thing that's missing from the myelin development field is a really good assay where we can look at the consequences of myelination and what that does to behavior. And so we are working on that manuscript right now. That's what I was doing last night, is telling exactly that story. The reason I'm not telling you about it at the cerebellum series is because we have not linked that to the cerebellum yet, but we can show that interfering with myelination in vestibular circuits disturbs that vertical navigation behavior that you saw. We can show you where in the circuit that's happening. And indeed, the zebrafish is myelinating at this time, and that has profound consequences for posture and balance. What it doesn't seem to have consequences for is the vestibulocular reflex. When we interfere with myelin, that seems to chug along just fine. So we've got all the elements that you just heard about coming together for a story about myelination that keep your eyes peeled. Thank you for that question.
Sure. So yeah, let's go to the audience. So Mikolaj, you have your hand up.
Hi, can you hear me well?
Yes.
I actually had closely related questions. So, when you mentioned a couple of those examples of animals that have balance skills and walking, running, is it possible to explain it by more by connectivity or more by myelination?
I think in those cases, if you examine the neuromuscular junction, you can see that develops much, much more in utero. So when they come out, the neuromuscular junction is quite well developed. If you look at humans and rodents and other mammals, it's not. And you can see that develop progressively. I guess if there's a cautionary note here, we could look centrally till the cows come home and see all kinds of interesting things that correlate with behavioral improvement without a way to measure what's really rate limiting. It's very tricky to pinpoint what's going on. And I think that's why it turned out to be very important to look out at the muscles and make sure, those really aren't even ready. And the rate with which they get ready seems to match behavioral improvement. So it helped us not get steered in the wrong direction. I would recommend for, I guess, looking at dexterity, that we be sure that the muscles are equally capable. And if so, is it indeed the myelin that's holding things back? I hope that answers your question, Mikolaj.
Salvo.
Thanks.
Thank you. Salvo, please go.
Good to see you.
Hi, David. Thanks for the very interesting talk. I have a question on the first part of the presentation where you had that nice correspondence between the maturation of the neuromuscular junction and then the behavior. Do you think that would apply as well to, for example, vestibulospinal reflexes, where perhaps there is a higher contribution from the muscle spindles as opposed to the vestibulocular reflex?
Right. So there in the zebrafish, at least in teleosts, maybe correct me if I'm wrong, but as far as I know, we don't see muscle spindles. There are muscle spindles reported in the jaw muscle of one teleost fish, and that's all that's ever been reported. So I don't think that that's going to be the source of feedback. So let's just put a pin in spindles. We have another story about vestibulospinal neurons. I think that the observation that the fish can twitch at 24 hours says that what's limiting postural development is much more likely to be central, not out at the muscles. So I think that the acetylcholine receptor stains are beautiful really early, and I don't think that would be rate limiting at all. I think it would be quite different. But I don't think it has to do with the muscle spindles per se, as much as the earlier and maybe evolutionarily determined development of trunk musculature.
Gotcha. Thank you.
Wonderful. Jean-Laurent, go ahead.
Hello. No,
Now I'm in trouble.
no, not at all, not at all. Thank you for this beautiful talk. And I'm, by the way, practically jealous of the zebrafish because they have a much nicer tilt vestibulocular reflex than humans. And on that topic, so do they have functional semicircular canal at the time?
Sure. So short answer, no. Long answer, it depends what you mean by functional. So the semicircular canals are present, they are wired appropriately, and on our TIPM apparatus, we can give a stimulus that's strong enough to elicit vestibular tuned responses in animals that are gravity blind. So the semicircular canals are going to be the only source of signals, so we're confident that that's where it's coming from. The lumen of the canal is exceedingly narrow at that age. So to just give you a sense, you could fit 3 or 4 of these fish under your thumbnail and you wouldn't know that they're there. So their canals are that much smaller and the lumen of the canal is that much smaller. So the force needed to displace the fluid in the lumen to create pressure at the cupula that would be sensed by the canals has got to be pretty big. So we can, under lab conditions, give that stimulus and show that the canals are working, but operationally the fish can't do that. And so it does not have, it has a lovely pitch and roll vestibulocular reflex, which is subserved by the utricle, but it has no yaw, so no left and right horizontal, even though the horizontal, there are lots of horizontal saccades, so nasal saccades, temporal saccades. The fish is making those all day long, but there's no yaw VOR, and that's does not emerge naturally till about 45 days when they, well, on average, when they reach a certain size, and that's when we think that canals come online.
I see. Thank you.
I hope that doesn't discourage you from studying the fish.
Oh, not at all. I mean, it's just telling me that everything you...
So many questions. I hope, I know we tend to be biased towards the canals because of historical reasons. But there's so much to do in the space of gravity sensation. So
of course,
please join me.
David, I thought one of the coolest things in your presentation was how you showed that when the fish become adults, they can move more like a helicopter. They can produce lift, rotating the pectoral fins, and then the coordination between this fin motion and trunk motion, presumably has some, as you showed, that's something to do with the function of the cerebellum. Do you think that there would be something like an ataxia in them that one could study by looking at the coordination between these different parts of the body during some form of goal-directed movement, like moving up?
It's interesting. So just a quick clarification. So they're older, they're juveniles when you start to see the coordination there. And adults, they're doing all kinds of even more beautiful things with their fins. So if you remove the fins, you don't see a net change in the elevation that they can achieve. It's not so much an ataxia, but they revert to the earlier mode of producing trunk rotations. So you can disrupt coordination, but the space of behavior that you see is just as populated. Does that make sense? That's it's the hesitation around what we would consider to be an ataxia model. I would defer to you if my understanding is that the space of movement isn't quite as broad in an ataxia space, and that's where you get the A, right?
You can make the movement better, but that doesn't mean you can't make the movement.
Yeah. So I think that would be the difference, is that because they have different strategies and different effectors, they can fall back on other ways to achieve the same goals. We're also looking at their natural behavior. We're not challenging them to move their fins in a particular way or giving them a stimulus that would cause a particular fin movement. I would have to think hard about how we might be able to do that. it's a really interesting idea. But yeah, for our initial forays, we have definitely opted to study the things that they do when we can't stop them from doing them. Maybe it's from training monkeys all those years, but it's much easier to not train them and watch them than it is to train them and hope. So,
Alexandra, please go ahead. We can't hear you.
Sorry, hello.
hi.
Good morning or good evening, depending on the time zone. A really beautiful talk. I actually have to say that now I am even more inclined to do something that I always sort of wanted to do, and to look at either literature or collect cerebella of mammals that can fully run and walk, like, within hours after birth. Because you showed, of course, these beautiful examples of, the rodents or us and monkeys. And we tend to really study these types of animals. But if you think of horses, right, they are fully operational, but in utero, they also don't have any gravity. So that really little sense of gravity, right? There's,
Yeah.
I would assume. So then the question I was wondering as you were talking is that, does this mean that their cerebella are just so fully developed that they can do something within like 10 trial learning that takes us, 10,000, 10 million trials? Or is this something fundamentally different in the way that
Oh, what an interesting question. What an interesting
because nobody studies like, like I would love to record like from a lamb of a
Yeah.
of a lamb.
What would, boy, this is the audience to ask, right? Does anybody
I know. Where can you ask this question if not here? Or next Gordon.
differential rates of motor learning, right? Are they, yeah, what?
I know it's very out there, and I know that this was all about fish, but this was just something that I was thinking about while I was listening to your talk.
What a great question. I don't know literature that challenge on maybe in the chick. Maybe in the chick that there might be early work on motor development. That's the place that I would look first, right? Because the chick has a embryological model, has a
And
lot
they're bipedal.
of right? And they're bipedal, but they have to be moving quite soon after birth, in the way that ungulates, right, would. So
Yeah.
that would be where I would look, but I really don't know. I really don't know if there's sort of motor learning differences there. Does anybody?
I'll check that, because I was just thinking about old literature, but then everybody used cats in the past, right? If you think of cerebellar recordings. So it's monkeys, cats, and of course rats, mice. But I wasn't, but chicks, that's true. There were, I will look it up. I'll look it up and then I'll get back.
Please let me know if you find something awesome. That would be really cool.
But yeah, I think this myelination story is also really fascinating. So thank you, and beautiful data.
Coming soon. I think I've taken up everybody's time enough. Thank you all so much. That was a lot of fun.
I loved it. Thank you so much. Thank you, everybody.
Be well.